Hello everyone, welcome to our discussion on non-allelic interactions or gene interaction, and in that we were discussing about the different types of non-allelic interactions, of which we came across complementary genes or complementary interactions which gave a modified ratio of 9:7, and we came across the second one, which was supplementary genes, where we saw the ratio, the normal Mendelian dihabit ratio was modified to 9:4:3. And in today's session, we are going to see yet another non-allelic interaction, which is called as epistasis. So, epistasis is also an interaction between two non-allelic genes, but here the interaction is in a special way where one gene masks the expression of the other gene. So here also two non-allelic genes are interacting together to produce a particular character or a trait. But when they interact, what they do is that one gene is masking the expression of the other gene, or it is inhibiting the other gene. So the gene that suppresses the expression of the other gene is called as the epistatic gene, and the gene which is suppressed is called as the hypostatic gene. So we will be coming across two types of epistasis. The first one is dominant epistasis. So a dominant allele of the first gene is masking the expression of the other gene. So the dominant allele of one gene, when it masks the expression of the other gene, other dominant gene, it is called as the dominant epistasis, and we will see the ratio is 12 is to 3 is to 1. And coming to recessive epistasis, in recessive epistasis, a recessive gene in the homozygous condition masks the expression of the other non-allelic dominant gene. So here we are going to see an interaction where a recessive gene is masking the expression of a dominant gene. So these are the two epistatic interactions that we are going to discuss, of which the first one is dominant epistasis. So here a dominant allele of a gene is going to mask the expression of the other gene. So we are going to take here the example of the flower, which is called as foxglove, and you can see here the three different phenotypes have been shown here. So here in this case, we are going to see the interaction of two genes which interact in a particular pathway, and that determines the color of the petals. So petal color, the trait is petal coloration, and we are going to see how two different non-allelic genes interact and produce this particular petal coloration. So here, what happens is one of the gene will affect the intensity of the red or the purple color. Red, it's not red actually. You can call it as purple. So the intensity of the purple pigment in the petal allele, in the petal. So allele small d, which is the wild type one, which results in the light purple coloration, which is seen in natural population. So this one, or this particular coloration, is brought about by the wild type allele, which is small d. So here you can see that as the notation given here is a small d, and this small d is considered as the wild type allele. So we have already discussed what is a wild type, so let's recollect what is wild variety now. So wild variety is nothing but the variety which is seen more in a normal population, or the more the number, uh, the common phenotype which is seen in a population. So that's the exact word to be used. The common phenotype that is seen in a particular population can be called as the wild type. So, there is a possibility that always we tend to use a dominant representation for a wild type allele. So, always the wild type allele may not be the dominant one in the, or the dominant allele in the of the dominant allele of that particular gene. The reason is that how we choose dominance in Mendelian, the same pattern is followed here. So, what is the particular phenotype that is expressed in an F1 generation when you cross two contrasting pure line plants? What you get there, that is called as the, that is considered to be the dominant one, and that dominant one with excess number will be represented in the F2. But that might be a mutant. We discussed about dominant mutations, etc. So dominant mutation is expressed in the mutant variety as well as in the F1. So what happens, or what the thing to be stated here is that do not confuse with dominance and wild type. So wild type does not indicate dominance. When you say a particular trait is wild type, it does not indicate that the trait is dominant. That trait can either be dominant or recessive. So if you see the case here, you can see the particular allele, which is the wild type allele, which is small d. So that one is a recessive allele of that particular gene, and that is causing the particular coloration, which is light purple in the natural population. And you have the dominant allele, which is D, which is a mutant one, and that produces the dark purple color. So this is produced by the dominant allele, which is the mutant one. And we are going to see the second gene. So this is the first gene, which is represented by two alleles, capital D and small d. The second gene is W, of which you have small w, which is a wild type, and that allows the synthesis of a pigment throughout the petals. So this allele, which is a small w, it causes the synthesis of the pigment in the petal. So these two are examples of small w, where when the small w is present, what happens is the pigment will be synthesized all through the petals. And if this other allele, the mutant allele, capital W is present, what happens is capital W is an inhibitor. When capital W is present, the capital W allele is present, what happens is this capital W is inhibiting the synthesis of either capital D or small d, or both. So if capital D is present in a plant, what happens is it will inhibit the production of that dark purple color throughout the petal, and it will be restricted only as spots that you can see here. If small d alone is present, the same thing happens, that light purple color will be present only as spots in the throat region. It is inhibiting the expression of this particular pigments, both the light purple as well as the dark purple. That means capital D as well as small d to express in the entire petal. And this will give rise to a ratio of 12 is to 3 is to 1. So this is the dominant epistasis. So here with this example, you can say that this W, or capital W, is the epistatic gene and D is the hypostatic gene. So W is inhibiting capital D from expressing. That's why we call it as a epistatic gene. W here is epistatic because that is the one which is inhibiting. Whenever W is present, capital W is present, the pigment synthesis in all other cells of the petal will be inhibited. Only the pigments can be seen in the throat region as spots. So here the inhibitor or the epistatic gene is capital W, and the hypostatic gene, the one which is being suppressed, is the D one, either capital D or small d. So we have justified this particular statement here. A dominant allele, in this case it is W, of a gene masks the expression of the other gene. Other gene can be the other gene. The both the alleles can be inhibited. Either capital D can be inhibited or small d can be inhibited. So this is what is called as dominant epistasis. So we can see the cross. So here we have taken two double dominant one, and this is the double recessive plant. So here the phenotype will be, you are having capital D and capital W. Because here you have a capital W, you can expect what will be the phenotype to be. The phenotype here will be, because there is capital D, we expect that it will have a dark purple colored petals, but the flower will be white. Because you are having W, the flower will be white flowers with dark spots. When you come here, here this is small w. As a result, the pigment will be synthesized in all regions of the petal, and the color of the flower will be light purple. So we are crossing these two plant, you get a dihybrid here. So in this dihybrid, we expect so here this capital D being the dominant one, it will have darker spots because W is present. the dark purple color will not be seen and through in the entire petal. You will have white flowers with spots. So this is and when you are cross fertilizing it, you have these the normal dihybrid cross. You have such a type of gametes. So here these are the four gametes, and you have the other four gametes here. This is F1 cross F1. In that, this nine, which is rounded in yellow, you can see 1, 2, 3, 4, 5, 6, 7, 8, 9. You have here 4 here plus 3 7, 7 plus 3 10, and 10 plus 2 12. So 12 is circled in yellow, and in that yellow you can see the presence of capital W. Whatever the gene DB, either capital D or small d, whatever it might be, because capital W is present, the flower will be white. The phenotype is white with spots. It might be dark pink spots, or dark purple spots, or light purple spots that might not cause any significance, but the flower is white. That's why we have a ratio of 12. You have 12 here, and you when you see this particular plant, you can see that they are having small w and the presence of capital D. As a result, it will be dark, or the flowers will show something which is called as the dark purple in color. The three one which is circled in blue shows dark purple colored flowers, and you can see the double recessive plant here. Double recessive, you have this uh, small d and small w. Because small w is there, you can see the pigment all over the petals, and small w causes light purple. And that's how you got the ratio 12 is to 3 is to 1. So what is happening here is, whenever this capital W is present, that is in this 12, in these 12, or this 12 is an example, or 12 spotted ones are examples of what is called as epistasis. In these 12, you have the epistatic gene, which is called as capital W, which is inhibiting the expression of either capital D. So here you are having all capital D's here, and here in these three, you are having small d. So this capital W is inhibiting both capital D, capital W is inhibiting both capital D as well as small d, so that the color of the flower is white. So this is what is called as dominant epistasis. Coming to the second one, which is called as recessive epistasis. So in recessive epistasis, what happens is a recessive gene in the homozygous condition is masking the expression of the other non-allelic dominant gene. So it is specific here. It is masking or inhibiting the non-allelic, the other genes, dominant gene is being uh, suppressed or being inhibited in this particular case. So here we can take the example. First, we will take two examples. First, we will take the example of supplementary gene itself. So if you see, you can see the ratio is same. That of supplementary gene as well as recessive epistasis is the same. That is 9 is to 3 is to 4. So here, when we are going to explain uh, recessive epistasis, and in recessive epistasis, we are going to explain recessive epistasis with these particular, or with these three individuals, we are going to explain something which is called as recessive epistasis. So if you compare supplementary gene and recessive epistasis, in supplementary gene we said that the gene called as A requires the gene which is called as A requires the presence of C for expression, but the gene C uh, does not require A for expression. The examples are can be seen here. So the example of that one is shown here. That means, as we said, this is an individual with only capital C here. So only capital C here, and C is expressed. It gives you a phenotype which is black. And when you're having both capital A and capital C, what happens is it gives a new phenotype which is a gouty in color, which is yellow or brown in color. A gouty color is expressed. So this is what is called as supplementary genes. That means when two genes interact, one gene is independent of expression, but the other gene is dependent. The other genes require the presence of the second gene. But when we are talking about recessive epistasis, we will be taking only these three individuals and explaining what is called as recessive epistasis. And in recessive epistasis, what we have explained is that recessive epistasis is the case where the a recessive gene in the homozygous condition is inhibiting the dominant allele of the other gene. So here we take the example or how this is acting or how they are inhibiting. So in this case of the coat color of mice, you can see that the gene C and A, they are the genes which encode enzymes that act in a particular pathway, that is for the production of the pigment, and the pathway is shown here. So in this particular pathway, you can see that you have a compound which is called as X, that will be acted upon by the enzyme that is produced by capital C gene. If it is capital C gene, the enzyme will be produced, and as a result you get the pigment which is called as black, and that is what you see here, these three individuals which is showing black color. So here they are having a capital C, and if you observe their genotypes, you can see that they do not have a capital A. The capital A gene is recessive. When the A gene is recessive, this enzyme will not be produced. As a result, all the three individuals remain black, because the other step in the particular sequential pathway does not function, or it does not move, or it does not the black pigment cannot be uh, converted to the agouti pigment, because they lack the three black individuals, they are lacking the enzyme which is called as the lacking the gene to produce the enzyme called as A. The capital A gene is absent in them, or it is recessive in them. As a result, they do not produce agouti. The particular pathway stops at black, so the pigment is black. So what happens in if both the genes are present, that is our nine individuals with a gouty color, when both the genes are present either in homozygous or a heterozygous condition, capital C will be there, capital A will be there, both the enzymes will be present, the black pigment will be converted to agouti, and that's how you get this nine individuals with agouti. In them, both capital A, capital T is present. As a result, they get agouti. Coming to our three of this four, we are talking about only three. The one which is double recessive, it does not have any of the. If you take double recessive, it does not have C, it does not have A. As a result, the color will be of this X, which is an uncolored pigment, which will be white or albino. So we are not considering the double recessive, but if you take this three, that is the other three of the group, you can see that they are having a dominant gene. So these are the other three. This one we are not considering in this because it is double recessive. Being double recessive, C is absent, A is absent, so that the pathway does not function in them. As a result, the pigment does not have any color, either black or the agouti color. The compound X is not getting converted. As a result, the color of the coat color will be white, or it can be called as an albino. But what is the case with these three individuals which are having a dominant gene, which is A? So they are having a dominant gene A here, but for A to act, okay, this particular A enzyme is present in them, but the substrate for the A enzyme is black pigment. The black pigment is not being produced in these three individuals. As a result, you can say that this homozygous recessive allele is, because it is present in the homozygous recessive condition, it is inhibiting the expression of this particular gene because C is absent. When small c is present, it does not produce the enzyme C. As a result, X does not converted to uh, the product which is called as a black pigment. So this is an inhibition which was happening in a step, or in a step which is in a sequence which is in a upper or a higher order than that of the normal reaction. Even though the gene is present, the A gene is present in the dominant condition, it does not have the substrate because substrate has to be produced by the C gene. As a result, you can consider these three to be examples of the recessive epistasis, where a recessive gene in the homozygous condition is inhibiting in the sense that because it is not there, the expression or the A gene, whether it produces the enzyme or not, even though it produces the enzyme, it does not have a substrate to act. As a result, it produces, or it will have a color which is called as, or a coat color which is called as the albino coat color. So this is an example of our the case with the coat color in rats, the same example of supplementary genes. And coming to another example in the Labrador is that uh, in this in this case of Labrador retrievers or dogs, there are two alleles for which is capital B and small b, now which stands for, the capital B stands for black color and the small b stands for brown colored coats. And there is another non-allelic genes which is interacting along with this B gene, capital B and small b. They are E genes, capital E and small e. So the allele small e is epistatic on these alleles, which is giving the coat color, which is called as the yellow coat color. So the two genes here in the case of this Labrador retrievers are the B gene as well as the E gene. So here the B genes, or the B gene is having the alleles capital B, small b. Capital B produces black color, small b produces is responsible for producing the brown color. And there is another gene like our C and A. There is another gene which is called as E, of which a small e is an epistatic gene, and that epistatic gene will is responsible for the formation of the color, which is called as the yellow coat color. And therefore the genotypes of these individuals or these dogs, whether they are having the capital B or small b in their dihybrid, where whether it is having capital B or small b, whenever small e is present, okay, whenever the small e is present, it produces the phenotype which is called as a yellow phenotype. And whenever capital E is present, if capital E is present, depending upon the B, whether it is capital B or small b, the color occurs. That means that the condition is that in this case, it will have a genotype which can be either, if I write the genotype of this black dog, the same thing happens. It is the interaction between two genes, and we are taking two different so here it can be either capital B, the genotype can be capital B, which is for black, and this should be definitely capital E and capital E. This is one possibility for black, two capital E's. The second possibility is it should be either capital B, small b. This also will produce black, and uh, it can be a heterozygote, capital E, small e. So in these two conditions, you can expect, and there's another one more condition, you can take it as this one to be heterozygote, black, and this to be homozygous, capital E, and the other one also capital E. So these three genotypes can give black color. So the same the case with brown color. When you have a small b along with capital E small e or capital E, you can of two genotypes, you get a brown color. Whenever small e is present in homozygous condition, the same thing will happen. That means in the if you draw a dihybrid, you can draw a particular dihybrid cross and see this three like these three individuals with the small e and capital B or small b, you will have the condition which is yellow coat color. So we can expect this to be 9 this to be 3 and this to be 4. The double recessive will also produce this particular color, which is called as yellow. So this is another example of recessive epistasis. So we, in this particular session, we discussed epistasis, dominant epistasis, and recessive epistasis. So recessive epistasis, we are taking the same example as that of supplementary genes. We explain recessive epistasis with these three individuals, how these three individuals, even though they were having a dominant gene, they were showing the phenotype of a double mutant. The explanation was that because of this homozygous recessive condition, this particular synthesis does not happen. As a result, even though capital A is present, it will have the feature of a double recessive feature, which is albino. So that's all with this particular topic, which is called as epistasis. In the next session, we will see two small topics, which is uh, polygenes, followed by transgressive variation and pleiotropism. Thanks for hearing.